Okay, we're recording. So I think most of you attended the last lecture, and if not, you can find some information about that. I'm going to post a link to the meetup group, and I just recently posted it to YouTube. So that should be available for those of you who haven't attended. So we're just going to briefly do a little bit of review about what we covered last week first. So last time we discussed how spike detection involves various hypothesis tests that attempt to separate noise from spikes. And so the very, very most general basic framework is that you have, some observed signal or signal vector. And the null hypothesis is that it is equal to just noise, right? So that would be, in this context, usually we're assuming that it's just going to be stationary, although that assumption is oftentimes broken in practice. And then the alternative hypothesis is that we've got a signal that's composed of both noise and a spike. And we saw that there are two major forms of noise: the electronic instrumental noise, which tends to just be, Gaussian, and then biological noise, which is where some of that confounding, where it can confound some of those assumptions. We also saw that these hypothesis tests use log likelihood ratios. So this is the sort of Bayesian theoretical optimum. You have some test statistic, and it's going to be proportional to the probability of y given h1 over the probability of y given the null hypothesis. And basically this form we'll see will just keep coming up, and in fact it does come up even when we discuss wavelets. And so the optimal estimator is just, okay, if it's less than gamma, which is some threshold that we set, then we retain the null hypothesis, and if it's greater than gamma, then we reject it. So that's fairly straightforward, and I'm not going to get back into the details of exactly what the tests were, but the main thing is that those were in the time domain. And we also saw that it's often useful and even robust to just rely on a simple voltage threshold instead of an optimal test. This has to do with the fact that oftentimes our assumptions for the various optimal tests are violated anyway. So we might as well just sort of, throw out, caution and just, do something that's cheap and fast. And one of the main reasons that we do that is because we, hang on, let me just quickly. Okay, so anyway, so it's often useful and robust to rely on a simple voltage threshold because we can, sort of, get a lot faster, cheaper computation. So this is often useful for doing computations on a chip that might be implanted in the brain, which oftentimes doesn't have a very large computational budget. We learned that spike centering is expensive and that window selection is problematic. We also learned that with unknown spike profiles, there is no universally most powerful test. We learned that estimates depend on the signal model. So if we don't know what the signal shape is, that oftentimes significantly reduces the efficiency of the test. And we also, at the very end there, learned that transform-based models often outperform time domain models. And we learned that the wavelet transform is currently by far the most successful transform-based method. So now we're going to get into what the wavelet transform actually is. And, it's used both for spike detection and for spike sorting. So that's why I thought it would be a good segue into the spike sorting topic today. The wavelet transform represents a function as a linear combination of basis wavelet functions that are generated from a mother wavelet, usually which are usually scaled versions of the wavelet. So here in figure 4.4 from So this is from another book. Let me grab the title. So this is from the book Brain-Computer Interfacing: An Introduction by Rajesh P. Nrual. And so that's actually a pretty good resource if you want to get a little bit of an introduction into the different types of wavelets and how wavelets can work. So the wavelet transform results in a sparse representations of signals as an orthonormal basis. So, basically what happens is you have these scaled versions of these wavelets, right? So you're going to have one that looks like this, and then you might have one that's like kind of, scaled to a higher time scale like that. And it turns out that for a lot of these mother wavelets, you can actually form any function, right? So it kind of works like a Fourier transform, where you can approximate any function using cyclical functions. And so the wavelet transform is a result, results in a sparse representation of signals on an orthonormal basis. So that basically what we have is y equals the spike plus some noise. And this is equal to some coefficients times this basis matrix, where only a very few CI values in C are non-zero. And so the wavelet transform is a linear combination of a small subset of the basis wavelet functions. And you can see here what that kind of looks like. So, you're going to have some distribution, and you're going to see that a lot of these are very, very near zero, whereas some of these other ones are significant. Like these are definitely, coefficients that you would want to retain. And the singular, and since phi is a singular positive definite matrix, its inverse is just equal to its transpose. And as a result, we just compute C by phi transpose times y, which is the original signal vector. And we can decompose this into C from the signal and C from the noise. The hypothesis test then becomes basically just the coefficients associated with noise for H naught. And for the alternative hypothesis, you have the coefficients associated with noise plus the coefficients associated with a spike. And the sparse nature of the wavelet coefficients implies that a very few large coefficients stand out against many very small coefficients that are near zero. These large coefficients usually demark sharp signal transitions, spikes. As a result, the coefficients below a predetermined threshold can be discarded, and the remainder will indicate spikes. So this is just an illustration of how that looks. It's taken from figure 2.5. And you can see how when you impose the wavelet decomposition, what you start to see is that there are these kind of outliers that stand out. They're sort of marked off in red here. And those are the ones that exceed the cutoff threshold, right? So we're identifying spikes from various decompositions in the wavelet transform domain. So we estimate the cutoff threshold by this formula here. So basically the absolute value of the coefficient must be greater than the standard deviation of the noise times the root 2 log n, where n is the number of coefficients in phi. And we compute the standard deviation of the noise with something called the median absolute deviation. And this underneath here, this number here is actually, it's theoretically a bit complicated to derive. But I've cited a paper at the end for people who are more interested in understanding why we use this particular divisor. But basically, this is just very similar to previous methods that we saw. The difference is basically that we're doing a hypothesis test in the wavelet transform domain rather than in the time domain. So the hypothesis test ends up taking the form, right? So we have this estimator here, y bar, and it's just y bar transposed times y bar. And if it's less than lambda, then we retain the null hypothesis. And if it's greater than lambda, then we reject the null hypothesis. And again, remember, lambda is just this log of the ratio of the probability of the null hypothesis over the probability of the alternative hypothesis in conjunction with some ratio of cost functions associated with falsely inferring the null hypothesis or falsely inferring the alternative hypothesis. So before the wavelet transform, the discrete Fourier transform, which decomposes functions into cyclical functions, was the main transformed domain for spike sorting. Unfortunately, the discrete Fourier transform has the same problem as the time domain, which is to say window selection. Picking a window size for the discrete Fourier transform entailed computing a trade-off between frequency and time resolution. By contrast, the wavelet transform tapers off about a central point of interest, circumventing the issue of window selection. So basically, as we can see from these illustrations here, you solve the window selection issue by just saying, okay, well, you know what? The magnitude here is just going to taper off. So I know that as long as I center my approximation about a region of interest, extraneous influences are going to vanish. And so that makes it really convenient because you don't have to pick a window size, which can often be a very arbitrary thing to do. So as you can see, this is very useful for spike detection. And we'll see later that it also tends to be very useful for spike sorting approaches. So does anyone have any questions about the wavelet transform method?
I don't have any question, but I have a comment. I've seen the wavelet transformation used in so many different domains, so many different sectors. And it's, I think, something that's still becoming even bigger with time. I think people are still flocking to it. So I'm looking forward to learning more in the future and maybe even using it myself sometimes in the future.
Yeah, it's definitely, it's become kind of a universal go-to domain, transformed domain to look at signals. And so in fact, there's another book that I have called, I think it's called Time Series for Neural Data Science, or Time Series for Neural Data. And basically that one's focused entirely on EEGs. But we also see that the wavelet transform comes a lot. So basically any place where biological signal processing is happening, wavelet transforms can be very useful. So moving on to spike sorting, there are two main approaches to spike sorting. The first one is pattern recognition, and the second one is blind source separation. The pattern recognition approach entails detecting spikes, extracting spike instances, and sorting spikes. On the other hand, the blind source separation approach tries to decompose the data from the signal array into a time series of n distinct sources. So basically, in one case, we're going in and we're basically detecting spikes and then doing a standard sort of context-free sorting, right? Something that isn't time series based. Whereas in the blind source separation, what we're essentially trying to do is decompose the time series into component parts. So the pattern recognition approach starts with spike detection and spike extraction. And so as we saw previously, there are three main methods for spike detection. An optimal time domain test that's based on the assumption that background noise is stationary, with a subsequent centering and window selection algorithm, which, as we discussed, is a bit expensive. There's also a simple voltage threshold method that uses a fixed one-size-fits-all spike centering and window size procedure. So that one, remember, basically what happens is that whenever a signal exceeds a certain voltage threshold, it just goes back half a millisecond and then pulls a window of length 1.5, right? So you end up with something where, like if you see a threshold kind of beginning to take off like that, it's going to cross the threshold here, then it's going to go back. And then empirically, they've found that if you just go forward a full millisecond from there as well, you're going to capture a nice portion of the spike. But the thing is that spike durations can actually vary significantly. And so this could be badly wrong. Although in practice, it tends to work out okay. And then the last one is a wavelet detection transform technique that circumvents centering and window selection altogether, which can be very nice. And it turns out it also, because a lot of it leverages matrix math in the discrete case, it can be fairly easy to implement on the cheap, although probably not inside of the chip that's implanted in someone's brain, at least not yet. Although, it probably would be something that that might be implementable in, like, say, a Neuralink chip that is, linked to the, to a module outside of the brain, but still on the person. So then the next thing that in the pattern recognition approach is we extract features of spikes. Most commonly, a dimensionality reduction procedure like principal components analysis is conducted. And so principal components analysis rotates the axis of a multivariate distribution until the covariances of new components are zero. And basically what we're doing is we're trying to maximize the variance, right? As you can see, if you pick these orthogonal components, we can maximize the variance, and that ends up being equivalent to making the covariances zero. And again, that's very useful because then you can start, doing, sorting procedures on data that really maximizes separability. So imposing a rotation that minimizes covariance is equivalent to maximizing the total variance. And amazingly, there's actually a closed form solution to this problem. So we simply need to solve for the eigenvalues and eigenvectors of the covariance matrix S. And we derive the characteristic equation for the eigenvalues using the formula where we set the determinant of S minus lambda times the identity matrix equal to 0. And so this results in a series of characteristic equations that when we solve, we can then plug in here in order to solve for the eigenvectors. And when we construct a diagonal matrix out of the eigenvalues, right? So this diagonal matrix here is equal to essentially lambda 1, lambda 2, lambda n, right? And then there are zeros on all sides, right? So we have this diagonal matrix here, and then what we do is we concatenate all of the eigenvectors, right? So W, on the other hand, is equal to essentially just like we've got, V1 down, V2, V3, etc., right? And this essentially, you can use this method to decompose the covariance matrix, the empirical covariance matrix. And then it turns out that the optimal rotation is actually just this eigenvector matrix, right? So we just apply it to the data matrix and we get a new matrix in the basis that maximizes separability. Now, if you were sort of astute, you would notice that this is equally applicable to the wavelet transform or any other transform domain as it is to the time domain. And so, it can often be very useful to do a pre-processing step that makes this, that accentuates shape factors over size factors when it comes to the spikes. PCA is popular for several reasons. One of them is that time domain spike amplitudes are poor discriminators. And so it's, we can capture shape-related factors in the PCA, and that can, additionally be accentuated by doing some pre-processing step, such as a transform domain. PCA leverages efficient matrix math, which reduces computational costs compared to doing optimization-based techniques. So a lot of similar techniques that you might do rely on gradient descent functions or some other iterative process that requires a tremendous amount of computation, whereas this one just requires matrix multiplication. And so that makes it kind of a go-to choice. The other thing is the PCA, as I've described it, the one that uses the covariance matrix, whitens the data, strengthening the stationarity of spike forms. And so that's actually very useful because you're essentially imposing a centering without having to do, a whole additional set of pre-processing steps. And that then, of course, because it strengthens the stationarity assumption, allows us to assume that the profile of a spike isn't changing over time. Because, of course, in this case, if we were to train data on certain spike profiles and then a neuron migrates or it changes its activation profile for other reasons due to learning, it would invalidate the model, right? So that reduces the need for updating, which is a very important piece for the pattern recognition type of process. And then after we do some kind of feature extraction like PCA, the next step is we do clustering. And so I didn't really get too deep into k-means clustering here because I think it's something we can look up, and it's fairly standard, fairly accessible to people, and it's really not the only method. One important thing to note about clustering techniques is that in general, even though they are unsupervised, they do require human supervision, right? Supervised learning just means that you have ground truth to compare to. And in fact, in some cases, you can get that ground truth. So one of the popular methods for obtaining something, and again, this is not strictly speaking 100% sure that it's ground truth, but if you want to do something a little bit more nuanced, you can actually have human beings tag different regions in overlays of neural spikes. And this tends to be fairly effective at establishing the difference between various spike profiles. And so oftentimes what people will do is they'll tag a subset of the data, and then they'll use that for something such as k-nearest neighbors clustering. So that is also a possibility. You can sort of do a supervised training algorithm and then use that to get your first approximation of your model. And again, remember, for all of these things, because it's either unsupervised or because you're relying on training data, you're assuming that the spike forms are not changing. So that can be a big disadvantage to this kind of technique. So just recapping the overall structure of pattern recognition workflow. Basically, what we're doing is we're starting with some form of spike detection, and then we're moving on to spike extraction and alignment. And then we're extracting some features. And then we are doing some form of clustering. And after that kind of cluster cutting or labeling, or after some other implementation of an algorithm, for instance, if we have some ground truth already established, we might use other machine learning techniques. We then classify the various events into different categories. And we assume that these categories represent different sources, different neurons that have generated these various spikes. So as I've been kind of hinting at, pattern recognition does have some significant weaknesses. One of them is that the feature extraction techniques such as PCA do require human judgment, especially if you're doing it for dimensionality reduction, because in that case, what you're actually going to be doing is you're going to take that eigenvalue matrix and you're going to essentially drop some of the zeros or some of the lambdas, right? So you might have, ordering from largest to smallest. You might keep like lambda 1 and 2 and so on. But then at some point you might say, well, these lambda values are too small. And so I'm not really getting much out of them. And it's just costing me a lot, maybe even adding some noise. So let me set those to 0, right? And when you do that, you get a sort of condensed approximation of the original data set if you were to recover it using the formula that we saw. And these kinds of judgments, there's no statistical test that says this is the optimal amount of energy, meaning the optimal number of lambda values to retain. And so that tends to be kind of an issue, because if you see a drift in spike profiles, you're going to at some point have to make decisions. The other thing is that the quality of pattern recognition depends on how well spike extraction was done. So as we saw, window selection can be a significant issue. And in the case of wavelet transform, that tends to be very useful, but there are still some subjective choices, such as which wavelet mother to use when generating a basis function. The other thing is again, I think I've been kind of mentioning this repeatedly. This method is vulnerable to the non-stationarity of the spike profiles. And this is something that's known to happen because of cell migration and metabolic changes. And so it's not something that can just be ignored. The other thing is that the reliance on spike shape alone can lead to distant but powerful neurons appearing to be the same as nearby neurons, right? Like if they have a similar spike profile shape, like imagine you have a distant neuron that has a big spike profile that looks like that. And then you have a nearby neuron that kind of has the same spike profile, but smaller. Those two neurons are going to appear to be the same thing. And that may not happen often in practice, but in reality, we don't have really a way of verifying that it does or doesn't. And given that there are billions and billions of neurons in the human cortex, it's safe to assume that it can happen sometimes. The other thing, and this is known to happen a lot actually, is that many spikes will overlap in the data set. And so this confounds pattern-based spike sorting, because you're going to have one window associated with two spikes. And it turns out that overlapping spikes often have important information about the neural code, because neurons that fire together wire together, right? And there's other reasons as well. But the main thing is that neurons that are firing nearby in time will oftentimes have an influence on one another in terms of learning. So it isn't something trivial that we can sort of ignore in the same way that we might consider ignoring something such as the possibility that distant neurons look like nearby neurons. And there is actually a technique to address this, and it's called blind source separation. So under some circumstances, blind source separation can ameliorate the issues with overlapping spikes. Blind source approaches assume that there are P neurons impinging on an array of M electrodes to form an observed snippet, Y. This snippet can be decomposed into a noiseless signal mixture, X, as well as an additive noise component. So X is a bit different. We're not assuming that there's just one signal in here in X. X actually is a mixture of a bunch of different signals. So imagine, you have a bunch of different neurons firing, and then you've got this electrode here, and they're all impinging on the electrode, right? So you've got these signals propagating onto the electrode, and it leads to this agglomerated signal. That's what X is. And X can then be further decomposed into a mixing matrix, A, and a neural signal matrix, S, right? So again, suppose we have these guys, right, which are all elements of S. Those are all, in S. And And then this guy here, as well as some other detectors, right? Those are going to be in X. And what we're basically saying is that there is some A, some signal mixture matrix A that converts the time series associated with all of these neurons that are generating signals into the mixture associated with these various electrodes that might be sort of sitting in the neighborhood of those neurons. Okay. And it's a little easier to visualize here with this illustration. So we've got P signals here, right? That's going to be P signals. And then we've got, N electrodes here. And what we're saying is basically that there's some mixing matrix that basically, each of these arrows represents impinging, right? And we can almost, if you really think about it, this kind of resembles a neural network, right? It's kind of interesting. And in fact, in reality, if you had like, some electrode sitting here in an array, right? And then you have a bunch of neurons kind of, living in between, okay? The signals are going to weaken, right, as the inverse square. Let me, you know what, I think this would be easier if I have two different colors. So let's say that those are the electrodes here. And we've got these neurons and glial cells kind of living, right? So basically, this structure, if it's big enough, is going to look kind of like a convolutional neural network, right? Because at some point, the influence of like a neuron over here is going to be fairly insignificant, on an electrode over here, right? So what we're going to get is a mixing matrix that kind of, if we were to put it in a neural diagram form, would be kind of like a convolutional neural network where, we have some influence propagating out to like a certain distance, and then it stops. And then, something similar, and so on, right? So that's actually, something that's interesting is the resemblance here. Now, the book doesn't mention anything about using neural networks in this context, but I have a feeling that if you used unconventional activation functions, you might be able to get something pretty interesting here. So I just wanted to mention that in case people are interested in trying to do their own kind of thinking and work on this topic. So the goal of blind source separation is to find the signal source estimates, which can be summarized in a matrix of signal source estimates, S hat. And if the mixing matrix A is known, then there is in fact a minimum variance best linear unbiased estimate, which is equivalent to this formula here. Now, this of course is not generally the case. We don't know what the mixing matrix is. Although we can make some assumptions about the mixing matrix, as I mentioned, based on the structure of the cortex, as well as the structure of the array. So it's not a completely lost cause to try to reconstruct it, but in general, we tend to assume that we just don't know the mixing matrix. And in addition to that, there are two circumstances that you can have for the blind source separation. One is where there are fewer sources than there are neurons. And the other one is when there are more sources than there are neurons, or I'm sorry, when there are more neurons than there are electrodes, right? So the sources are, in this case, the neurons, and the mixtures are the electrodes that are receiving the impinging data. And so what we're saying is, that there are fewer neurons than there are electrodes in one case, and then there are more neurons than electrodes in the other case. Now, obviously, the first case never happens, but as we'll see, there are some ways around it, so it's useful to at least think about. So when there are fewer sources than mixtures, the most popular approach is independent component analysis. And in this case, basically S can be estimated by iteratively finding the linear transformations such that S hat is equal to W times X. So, I don't want to get too deeply into this, but the main point is that if there is no noise, then it's provable that this estimator algorithm will asymptotically converge to the true source array. Now, of course, this assumption is almost never actually true, right? There are almost never fewer sources than electrodes. In addition, it also assumes that the neural signals are independent, which is almost never true. And it assumes that only one source, the background noise, is normally distributed, and that the noise signal is fairly small. Okay, now, if you're familiar with PCA, then you might have kind of noticed that these first three assumptions can actually be resolved by imposing a PCA, principal component analysis on this, right? If we rotate the basis and then impose dimensionality reduction by dropping some of the eigenvectors, we might be able to get it so that P is less than N, right? It may not be the case, but in a lot of cases, it could be that we could do this. We could deliberately drop at least enough data so that we get P less than N. And of course, it forces the components that result to be independent. However, the size of the noise is something that has to be addressed at the experimental level, right? So, while we can resolve a lot of these issues with PCA, we would have to take special care to reduce the noise at the outset of an experimental procedure. Otherwise, this method wouldn't really be efficient. Now, in the case where there are more sources than mixtures, in other words, there are more neurons than there are electrodes, which is always the case. It's generally impossible to recover a p-dimensional source vector from an m-dimensional mixture in that case. However, if we assume that the source vectors are sparse, then an estimate of S can actually be recovered. And, as we saw, usually spikes, if we're just talking about the spikes, are in fact sparse. So if we get rid of noise in some way, then this is actually not an unreasonable assumption. And so there's a technique for looking at the signal subspace and trying to basically reconstruct the signal subspace when we have the sparsity condition. It's called multi-resolution analysis of signal subspace invariance. And we express the signal matrix here as S equals C, which is a big matrix of coefficients times phi. Now note that it's not an accident that the notation looks like the wavelet transform. The reason for this is because, of course, the wavelet transform does produce sparse representations, and it's probably one of the best ways to get a sparse representation to use for this method. Although theoretically you could do other things as well. So basically, it looks the same as the wavelet transform. We've got these, phi component basis functions, and then we've got these coefficients. And what we're assuming is that K is greater than P, meaning that the dictionary size, right? So essentially the basis here is much larger than the set of coefficients. And in the noise-free case, it turns out that the estimator y bar is just equal to the mixing matrix times this C times phi. And when the subspace C is sparse, meaning that so Kp would be just the set of significant coefficients, right? So if we have a vector of coefficients here, say because we did a wavelet transform, we're gonna have a bunch of zeros, and then there's gonna be like some coefficients, sitting among the zeros. And, we're going to have just a few of these, right? So that's what this is. This is the set of those. And if it's much smaller than the size of the basis dictionary, then the singular value decomposition of y bar can be computed for each non-zero entry. And so then, you get this formula. Again, this is pretty straightforward. The singular value decomposition is very similar to principal component analysis. It's just an adaptation of the same idea, except in this case, we're doing it with a matrix that may not be a square matrix that is symmetric. And so this makes it possible to do that. You can look up more information about it. It's pretty standard stuff in statistics. So I didn't get too deep into it. And basically, the columns of the eigenvector matrix UK span the column space of y bar sub k, right? So for each of these coefficients. And as a result, we can sequentially estimate the signal subspace spanned by the columns of the mixing matrix. Now, there's an algorithm for this that OIS produced and talks about in the chapter. I didn't get too deep into it, partly because I kind of ran out of time yesterday when I was kind of working on this, and also because I don't really want to misrepresent his method. However, if we want to go deeper into it when we get into trying to implement these algorithms, we can maybe do something where we put our heads together. So moving on to the conclusion, what are some constraints in open problems? So as we've seen over the last couple of lectures, there are several key constraints on real-world neural implants. One of them is that wireless communication from a chip is limited, so on-chip data processing is desirable. And so as a consequence, efficiency is often very important. So these algorithms, it's not just about finding the optimum, the way that you might do in like big data science, where you have like, a supercomputer or a cloud array that you can just throw at any problem. You're going to want to find really fast and clean techniques that also make a fair trade-off with the optimality of the estimators. The other thing is that as electrosize scales, viable algorithms are going to need to scale linearly or sublinearly in order to remain practical. There's also significant interest in leveraging the discrete wavelet transform methods and sparsity assumptions to enhance detection and sorting, right? So OIS, for instance, develops this sensible signal models that assume the blind source separation coefficients follow a power law distribution. And that's one example of a way of tackling this kind of open problem that exists in spike sorting and spike detection. So these are some practical notes that OIS left us with. One of them is that focusing on the classification of spikes oftentimes outperforms the attempts to reconstruct the entire spike form. So think about the idea of just, attributing spikes, whatever they look like, to various sources, right? That's one way of thinking about things. And you're not too worried about whether they exactly, if we're able to exactly recover the shape of the spike and if we know all the special details and so on. Now, obviously, for some cases, we might be interested in that for other reasons, right? Having to do with the study of the physiology of neurons. But if all we're trying to do is ultimately generate spike trains, which are these kind of point processes, right, over time, then it actually pays to just focus on separation and not worry too much about what the spikes look like. Another thing is that sensory and behavioral correlates can be used to enhance spike sorting. So in our last discussion, we had a guest who talked a little bit about her work doing research at a vision lab. And what they tend to do when they look at neurons in reality is they will differentiate types based on information that they already know to be true about, how different kinds of neurons behave in response to various, in this case, visual stimuli. And so if you're actually doing this in practice, you're going to have a brain region, and there's going to already be some research usually on what kinds of neurons exist there and how they differentiate based on different kinds of stimuli that have been presented. And so if you're doing some kind of training or a calibration algorithm, one thing you can do is do it under experimental circumstances where you present the subject with various stimuli designed to sort of maximally differentiate the activity of different kinds of neurons under different circumstances. So in practice, we actually have a lot more resources than have been discussed in this book. And that's something that might be interesting to explore when we move on to implementing spike sorting. Another thing is that trial-to-trial variability in neural behavior may have a significant contextual and deterministic component. However, decoding neural coordination is still beyond current methods. So this is kind of an up-and-coming thing, and this was back in 2010 when it was beyond current methods. So it's very possible that that new methods have emerged. And so another kind of open problem, or really just a thing to keep an eye out for, is what are some deterministic models of how neural spikes change over time in response to environmental factors, and how do neurons coordinate as a group, and what are the deterministic components of that kind of coordination? Another thing to note, just from a practical perspective, is that decoding neural signals without spike sorting works poorly at short time intervals, at least with current methods. So it's not a great idea to take a raw neural implant feed and just like stick it into a machine learning algorithm. It tends not to actually do so well, even though that approach has worked surprisingly well in a lot of other domains. So that's actually kind of promising. It suggests that we know what we're doing and that spikes are an extremely important component to the actual way that the brain processes information. Because if that weren't the case, then we might find that sort of completely ad hoc blind approaches might work fairly well. And as a final takeaway, establishing ground truth of neural spike trains is highly non-trivial. So don't take the pre-processed data for granted. This is something that almost every neuroscientist I know does do. They take this for granted. And it turns out that, in fact, there are a lot of subjective choices that go into generating neural spike trains. And so it's really something that people should focus a little bit more on, because there's a kind of an upper bound to the technical progress that we can make without really making sure that our ground truth assumptions are something that everybody in neuroscience who deals with spike trains is familiar with. So that's all I have for today, and I'm wondering if anyone has any questions about the second half of the lecture.
Once again, not a question, but what you were saying about notes. The notes, something that I wanted to comment on when you were talking about deterministic models for how the shape of spikes change. Mm-hmm. So that stuff I know often comes down to molecular dynamics. So sometimes that's things like if a neuron is firing a lot, then maybe the ion concentrations extracellularly relative to intracellular will change because it's been firing so much. Also, all sorts of things with neurotransmitters, and there's sort of all these dynamics around short-term plasticity. So how a neuron might be changing over the seconds. For example, neurotransmitters in an axon terminal could get depleted, and that might, I guess that changes, well, that changes some things. But there's a lot of dynamics like that where that have been mapped out pretty well. And I'm not remembering the name of a lot of them off the top of my head, but those deterministic models are out there, and those have been, there's been a lot of ground in that field covered over the last couple decades.
Awesome. Thank you for sharing. Let's definitely, I think that's something that's worth maybe looking up a little bit for future, spike train related models.
Yeah, maybe since next week I'll be covering the biophysical modeling. Maybe that will come into biophysical modeling.
Yeah, that's great. It looks like there's a very nice synergy here emerging. All All right, so if people don't have questions that they want recorded, I'm going to stop the recording now so that people can discuss more freely. Okay, we are no longer recording. Oh, actually, I'm sorry. I pressed stop share.